Good afternoon, and welcome to the PRISM e-Symposium. I'm Patricia Sampieri, and I will be your co-moderator today for this session. Rachel Friedman will also be serving as a co-moderator. She's working behind the scenes and moving our slides. Rachel, please say hi to everyone.
Hi, everybody. Thanks for being here.
Thanks, Rachel. So this session is on chemistry. It's a concurrent session that runs from 5 to 5:30 p.m. We will have a second session on technology from 5:30 to 6 p.m. During the students' presentations, if you have questions, please use the Q&A button to write and submit your questions, and I will then select questions at the end of the presentations if time permits. Okay, so let's get started. Our first presenter is Jahvon Johnson. Now, Jahvon's presentation is the development of non-precious metal catalysts using organic chemical synthesis. Take it away, Jahvon.
Thank you very much, moderator. Slide 2, please. Okay, so aldehydes and ketones are components of a variety of industrial reactions that produce everyday materials such as perfumes, dyes, pharmaceuticals, and plastics. Because of this, it would greatly benefit society if these reactions that involve aldehydes and ketones could be both faster and more efficient. The most common way to do this is by using catalysts which form reactive intermediates, which lowers the activation energy of the reaction without being consumed, allowing it to happen faster. Next slide, please. This brings us to coordination or metal complexes, which are commonly used as catalysts. They're formed when a metal atom forms coordinate covalent bonds with a ligand. Ligands can be any molecule or functional group, and their size determines the size of the metal complex. These complexes can be as small as methyl groups bonded to a metal atom, or can be quite complex in the case of multiple ligand molecules being bonded to multiple central metal atoms, forming what is called a coordination polymer. Transition metals in particular are popular metal atoms because their partially filled d orbitals allow them to form multiple coordinate bonds with ligand molecules. Next slide, please. The goal of my research is to develop cost-effective and efficient metal complexes for use in industrial reactions using abundant earth metals. In order to determine the structure of these complexes, they must be crystallized and analyzed through X-ray diffraction. As many of you may know, crystalline compounds are composed of parallel rows of atoms separated by a certain distance. You can take advantage of this by using radiation with a smaller wavelength than this distance, which then enters the crystalline structure and diffracts. The diffraction patterns can then be used to elucidate the crystal's general structure. Because of this, it is important to develop high-quality crystals, and there are different methods to do so. Next slide, please. Here are the reactions and compounds that I'll be forming complexes with. They are N-phenyl-O-phenylene-diamine and 3-ethoxy-salicylaldehyde to form the first ligand, and again, N-phenyl-O-phenylene-diamine, but with 2-pyridine carboxyaldehyde to form the second ligand. For both of these reactions, I use cobalt and manganese as the central metal atom. Next slide, please. The first method of crystallization is the slow evaporation method. This method is fairly simple, involving adding the ligand and the metal, both dissolved in a volatile solvent, into a test tube. Over time, the solvent will evaporate and crystals will slowly start to form. For best results, evaporation of the solvent should take place for weeks or even months. Next slide, please. The second method of crystallization is the slow diffusion or layering method. This method is more complex than the previous one. As the name entails, it involves having 2 or more distinct layers of solvent containing the ligand and the metal being used. For this method to work, you must absolutely make sure the solvents do not mix during the setup, and that can be done by pipetting slowly. Over time, again, usually weeks to months, the solvents will mix and crystals will form. Next slide, please. For an example of how crystals are formed, here are some examples of crystallized compounds. Note how they aren't as particularly neat as you would normally imagine the crystals to be, and they can also have a fair amount of variation in the way that they crystallize. Next slide, please. As for my results, I have successfully gotten crystals, but when analyzed through X-ray diffraction, they did not diffract. This means that there was a problem with the crystalline structure itself, which is usually caused by impurities. Comparing the two figures on the left and the right, you can see the differences in characteristics between a good and bad crystal that impurities can cause. Impurities can cause large amounts of crystals to grow, but each individual crystal will be of much lower quality, while a good crystal will be a singular crystal of much higher quality. Some of you may have thought that having a lot of crystals was a good thing, but that is not actually the case. You generally want to aim for having one defined crystal with an excellent crystalline structure. Bad crystals also grow much faster than good crystals, but have a much lower chance to diffract when analyzed. Next slide, please. My next step in my research are to continue using these crystallization methods to form their appropriate complexes. If I am successful in doing so, then I will try using copper as essential metal atom due to having similar properties to the other two metals and being a cheap, abundant earth metal. Once these crystals diffract, they can then be tested for their ability to catalyze different industrial reactions, such as hydroboration and hydrosilation of aldehydes and ketones. Next slide, please. And thank you for having me.
Thank you, Jahvon. Having a few technical issues there for a minute. Sorry about that. That was very interesting. And we do have a question for you. Someone would like to know, can you make similar catalysts to degrade plastic instead of synthesizing plastic?
There are a lot of different catalysts for a lot of different industrial reactions. I don't know the particular reaction to degrade plastic, but there's most likely a catalyst for it, yes.
Okay, well, thank you very much. Okay, we're gonna move on to our next presenter, Kedwin Gabriel Melendez. Hi, Kedwin. Okay, Kedwin's presentation is Chiroptical Analysis of an RNA Hairpin Catechins. Just as a reminder, if during the presentation you have a question for Kedwin, please use the Q&A button to write out and submit your questions. Okay, Kedwin, take it away, it's all yours.
Next slide, please. Thank you. Okay, so my research was based off of two studies. The first study was where they used catechins and were trying to determine how they bound to RNA sequences and how this modified their levels in liver cells. The way that this experiment was conducted was by using two different grape extracts to bind the catechins to the RNA sequence, then using hepatic cell lines to determine their expression. The results demonstrate that the effect of the mRNA expression depends on the polyphenol chemical structure. The ability of the catechins to bind to the RNA sequences was measured using HNMR. Next slide, please. So my second study was about how DNA and RNA were being bound to targets of green tea catechin. The second study is about how T catechins can be bound to DNA and RNA. The experiment was done using surface plasmoid resonance assay in cold spray ionizing mass spectroscopy. By using these experimental techniques, it was determined that ECGCG, which is the catechin that they were studying, bound to both DNA and RNA. The significance of this is that it can be used to learn about cancer prevention. Next slide, please. So what are catechins? A catechin is a substance that is found in tea, wine, dark chocolate, nuts, and berries. And research shows that catechins act like antioxidants in the body and can help fight diseases like heart disease, diabetes, high blood pressure, different types of cancers, even to Alzheimer's disease. And some examples are epigallocatechin gallate and epicatechin gallate. Next slide, please. For my experiment, our samples of catechin and hairpin were provided by Dr. Lisette Delgado Cruzada. The RNA sample was then dissolved in water at 8.2 millimolar concentration, and this sample was run through a circular dichrosome instrument. Afterwards, different samples of increasing amounts of catechins, which were added to the RNA solution, were run through the same CD instrument. The concentrations of their sample were RNA by itself, and then ratios of 1 to 1.0.3 RNA to catechin, a 1-to-1 ratio, a 1 to 1.6 ratio, and so on. As the samples are run through the CD instrument, UV and CD spectra are recorded of each sample. So basically, the main premise of the experiment was to titrate nucleic acid with increasing concentrations of catechins and then see if there were any variations in the CD signal of the RNA. Next slide, please. So what is cyclodichroism? Cyclodichroism is a spectroscopic method that looks at the response of a chiral molecule as circularly polarized light goes through a compound. The way that CD works is that a polarized light will be shone at a compound. The compound will then absorb right and left circularly polarized light. Next slide, please. Then there will be a measured difference in absorbance between the right and left circularly polarized light as it passes through the compound being studied. This difference in absorbance is depending on the chirality of the compound. Next slide, please. This is different from UV vis, where you only need a chromophore. For CD, you need both a chromophore and for the molecule to have some form of chirality. Next slide, please. So the results demonstrate that when there's a concentration of equimolarity of catechin galley, which was the compound that we used, to RNA, one can notice the appearance of a weak negative band between 310 nanometers and 350 nanometers, as well as a increase in the CD maximum at around 275 nanometers. There's also an increase of intensity at a 240 negative band when there is too much catechin galley in the sample. Next slide, please. So these are, as I already told you the results, our next step would be to study other catechins and other concentrations to see how they would affect the CD spectra. Thank you. I would like to thank the audience for their time, Dr. Delgado for her samples, and the PRISM staff for their assistance.
Thank you, Kedwin. Another very interesting presentation. And you too have a question. Your question is, what other techniques could you use to validate your CD results? For example, NMR or IR, for example.
We used our CD instrument, did both UV and CD, and we were using the UV portion to validate, to make sure that there was an actual signal where the signal was.
Okay.
So that's why we have both spectras.
That makes sense. Thank you very much, Kedwin. Again, great presentation. All right. Our next presenter is Zixuan Mo, and Zixuan is going to talk about, let me just, a little difficulty again, here we go. She's going to talk about the design and synthesis of new nitrogen-rich organic ligands for metal catalyst development. And once again, I'd like to remind everyone, if you have a question during the presentation that you'd like to ask Zixuan, please just type it into the Q&A, and time permitting, I will ask your question. I will pick at least one question at the end of the session. Thank you very much. Take it away, Zixuan.
Thank you so much. Good evening, everyone. Would you mind to go to the next page? Thank you. And I would like to talk about the symmetry of a new ligand, and it will be part of the base metal catalyst. The next page, please. Thank you. The metal catalyst is extremely important in industry application, and in our lab for developing a new base metal catalyst, we will first use the ligand disease, and once the ligand products, we will use that to combine with the metal salts to produce our PRISM complex. The PRISM complex brings tiny in 3 atomers co-partner seat is in marrow donor configuration, like the one of the figure you can see on your left side, and those are the 3 nitrogen atoms sandwich metal ions in a like a cramp. So once the PRISM complex exists, we will need to evaluate the composition of this PRISM complex, and a good of the catalyst should have a higher percentage of the atom economy. It means a higher percentage of the discharge product and a few of the byproduct. And I use this ligand disease from my mentor as a reference and doing all my work. Next one, please. The next page, please. Thank you. And for my ligand is the types of the 135 textine and the 3 of nucleophilic environmental substitution relation occurs in this relation. And the temperature is a significant factor to control the yield of the product. Next slide, please. So there are two parts of the work I did. For the part one, I use camera starting compounds, the cyano chloride to do three of the chlorophyll substitution relation to get my crème product of the ligand. And for the part two, I mix the metal salts layer with the ligand layer to form the single crystal. After the single crystal coming out, the structure of the metal complex is analyzed by the X-ray differences. Next slide, please. Then I got the results. So for the part one is about the crème product of the ligand and is slightly yellow or the brown yellow sticky solids. The percent yield is around 30% to 69%. But however, because the solid is easy to absorb the water, so the real percent yield may be lower than the absorption. Next slide, please. Around a month later, I got a single crystal from my layers mixture and is on the right bottom on the slide. The green point on the structure indicate the copper ion and the blue point on the structure indicate the nitrogen ion. The structure is shown as the PRISM complex. So for this ligand, the percent yield of the cream product are very various. So due to the unstable yield as well as only the one of the structure of the compound salt complex was successful analyzed. So we will still need to develop it more PRISM complex production. Next slide, please. So for the future direction, we will evaluate the existing case, start the properties of this ligand, and modify the procedure. And we will evaluate the catalyst cavitation. And also we will adopt a similar method to other as-tracking diseases. The next one, please. So finally, I want to say thank you for my mentor, Dr. Zhang, and thank you for the word for encouragement and for all you are telling me. Thank you so much.
Thank you, Zixuan. It sounds like you have a lot of interesting work ahead of you, and this was a very interesting presentation as well. And you too have a question.
Okay.
Back too. So I'm gonna take the first one here. Besides being catalysts, do the triazine molecules you have synthesized have other potential uses?
Yes, other than being the catalyst, because for this ligand and also because it is the structure for the 135 textine, and because this textine have a higher ability to absorb the water, so we may consider developing that as a water absorption agent.
Very interesting. Okay, well, thank you again, Zixuan. And now, for our final presenter in this session, we're going to switch to Nicholas Towler. Again, I want to remind everyone about the Q&A button at the bottom of your screen. So fill out that if you have a question, as some of your colleagues have. And Nicholas will be talking about the synthesis and characterization of DNA adducts formed by a mitomycin C derivative. Take it away, Nick.
Thank you very much. Slide 2, please. Today, there are many drugs used in chemotherapy that can signal for tumor cell death adequately, saving a person's life. However, in addition to killing cancer cells, these drugs may also kill neighboring healthy cells, which may make the side effects of these drugs more unpleasurable and may have harsh consequences on the health of the individual undergoing treatment. A popular drug being studied right now for its cancer-killing benefits is mitomycin C. This drug elicits its effects by inducing DNA adducts, primarily the interstrand crosslink. What is an interstrand crosslink? Well, imagine the drug in between two strands of DNA. What the drug essentially does is grab onto each side of the DNA so it cannot go through its normal processes, such as replication. Since the cell cannot replicate, it signals for cell death, and thus the cancer cell is dead. Considering this same mechanism, if we killed tumor cells the same way as mitomycin C, but modify the drug just to be a little more selective towards the site of action, then the drug would kill less neighboring healthy cells and it would be more potent. The individual treated would feel less of the unpleasurable effects and would have a higher chance of survival. Next slide, please. My colleagues have developed a new hybrid drug given the temporary name DELS-15. This drug combines the DNA alkylating benefits of mitomycin C, however, contains a group commonly found in lexotropsins, which has an affinity for the minor groove of DNA, which is where these drugs primarily attach to. Next slide, please. In preliminary studies conducted by my colleagues, DELS-15 has been shown to be more cytotoxic than mitomycin C and has been shown to be more effective in producing the interstrand crosslink. In order to publish this evidence and to verify future in vivo results, we need to create the interstrand crosslink and then isolate it. This has been done with other drugs by biomimetic methods in which the drug is reduced and combined with small DNA sections called oligonucleotides. We have performed many of these reactions, changing the reactants slightly each time to try and optimize the synthesis of the interstrand crosslinks. Next slide, please. Biomimetic synthesis uses sodium dithionite to reduce the drug, enabling it to bind to the DNA. A second approach we have is trying to react the precursor drug, mitomycin A. The synthesis of the mitomycin A crosslink uses a similar protocol, however, it requires an extra step to convert the mitomycin A adducts into the DELS-15 adducts. Next slide, please. The DELS-15 biomimetic synthesis reaction mixture was enzymatically digested and then separated by high-performance liquid chromatography, or HPLC. These reactions showed a peak indicating that the crosslink was formed, however, in quantities too small to isolate. The crosslink was able to be generated in DNA with a central nucleotide sequence of CG and not GC, which is similar to that of mitomycin C. The mitomycin A biomimetic synthesis reaction mixture was also enzymatically digested and separated by HPLC. The chromatogram showed a peak larger to that of the DELS-15 reaction, indicating a higher yield. The crosslink was able to be generated in DNA with a central nucleotide sequence of CG. Next slide, please. This higher yield may be due to the fact that mitomycin A is more readily reduced. Therefore, it may be easier to link a larger amount of DNA to drug to DNA using biomimetic methods. We may have not found the right conditions for DELS-15 to efficiently link the strands of DNA, and this may be due to the fact that DELS-15 has many different properties as compared to mitomycin C. Although DELS-15 is a derivative of mitomycin C, the properties of DELS-15 are much closer to that of mitomycin A. Our future work includes finishing the conversion of mitomycin A adducts into DELS-15 adducts, changing the solvent system of the HPLC to a system that better fits with the drug's properties, and also generating the crosslink on a larger scale to have enough for future experiments. Next slide, please. I would like to thank William Aguilar, Owen Zacharias, Marian Romain, and Luisa Zuluaga for their guidance and support throughout the project.
Nicholas, thank you. Another excellent presentation. And wait, we got a question coming in. Give me a second. All right. Why did you choose to use mitomycin A instead of mitomycin C? So you use a derivative, correct, of C? Mm-hmm. So I guess they're asking, why did you use the regular A versus the regular C?
So when I was doing my literature search, I found a publication which used mitomycin A adducts and converted them to adducts of a different drug. So we decided to try a similar method with our new drug. And since in the synthesis of DELS-15, mitomycin A is only one step away, so you could imagine that they have very similar properties to each other.
Okay. All right. Well, you guys did great presentations and you came in right on time. So that concludes everything for this session on chemistry. I'd like to thank all of our presenters for their hard work and their great presentations. And I'd like everyone to give you all a virtual round of applause. Our next session is going to be on technology. That's going to start at 5:35 in the same room. If you'd like to stay with us, just stay in the room. There are other concurrent sessions going on different topics. So if you'd like to look at some of those topics and perhaps go into one of those rooms, please go to our website, which is www.jjay.ca. cuny.edu/PRISM, PRISM, and then follow the link to join the session of your choice. Thank you again, presenters, and thank you to our audience for attending. Bye-bye.